Okay, there we go.
Now?
Now it is. Okay.
Okay.
Sorry.
Hi. I'll tell you a little bit about my proposal, and then I'll take any questions. So the proposal has 3 parts. There's the 2 equipment pieces and the curriculum piece. The 2 equipment pieces are a autoclave, which would help sterilize all of our equipment, and a shaking incubator. The autoclave would reduce our consumption of plastic goods also, so we'd be able to spend more money on buying extra stuff for the kids to sort of experiment with, to design their own experiments, and they'd have not only the equipment, the petri dishes, microtubes, beakers and stuff, but they'd have more, reagents and chemicals and bacteria. So that's the autoclave. The shaking incubator allows us to keep primarily bacteria at a particular temperature, and it shakes at the same time because you can grow bacteria at larger quantities and faster. If you aerate them, they won't die. The incubator would let us grow more than a class's worth of bacteria at once, so the kids can actually use bacteria as the model organism it's great for. Because right now all we have is a traditional incubator, so it just keeps it at a particular temperature. The most exciting part of my proposal is actually the curriculum part. They're based around synthetic biology, which is allowing students to explore this new area of engineering, or biological engineering, where they can take different components, things like genes, promoters, regulatory parts, mix them together in different ways, see how they interact, and insert them into a living organism. The curriculum itself has 3 structured units, but I think once we figure out the parts of those, we can take them apart and play with them more. The first one has them experiment with bacteria, 5 different strains of E. coli. They put the same DNA code into all 5, but it turns out because each of the bacteria read it differently, interpret the chassis around the code differently, they come out all different colors. So the kids have to figure out, okay, why are they coming out different colors? They try playing with the different parts, they take just the gene, put it in. They take the gene and some of the regulatory stuff around it, put it in, to see if they can get them to become the same, or if there's something different about the interpretation that makes them all different. So that's one. They get to play with those pieces. Another one, they call it iTunes or something. They use different codes and different regulatory parts, this time in yeast. Similar thing, the bacteria come out in different shapes. It causes them to grow sort of in different patterns because of the regulatory proteins. And the last one, that smell, is a bacteria with genetic components that cause it to smell like bananas instead of the typical gross bacteria smell. The kids have to figure out why they don't always make the smell at the same time. It turns out the regulatory components there only turn on the gene at different cycles in the bacteria's life stage. And they have to figure out, well, how can I change this to express it when I want it to? So they get to explore with different ways of manipulating really genetic components, which is something you typically cannot do at the high school level. So that's my grant. Any questions?
I have a quick one for you.
Yeah.
DNA, there was an opportunity for adults as opposed to just students to have an opportunity to try this as well.
Yeah.
Is this something that could also be done that way? Is there a way to run something? I'm thinking about in Wellesley this coming Saturday or the Saturday from now. They're having this huge symposium that is for the whole community. Can this be used in a smaller way? So you're buying these wonderful, the autoclave and the shaking incubator. Can they be used for something other than this particular curriculum that you're thinking about?
Yeah, I mean, there's science components. You could use them in any of our science classes, the biology class does an independent research project, or the design project that deal with bacteria. We could do some sort of outreach to our community, either with parents or something in the summer, that could involve using these components. Because we would be able to reuse things, we'd have a lot more flexibility in what our budget could support.
The autoclave, I'm sorry to jump in, but the autoclave could be used physics through senior science.
Building, right?
Yeah, I mean, it would need to take sort of priority with the bacteria related things, but I mean it could be it's a tool that could be beneficial across the entire science curriculum.
How big is it in actual size?
The one that's here is tabletop size.
So you'll be able to find a spot for it.
Are there any ongoing, for either of them, I whether bacteria costs a lot of money.
You think it would be free, right?
Kind of E. coli you could find at the store, but are there any ongoing costs that this would?
For the two big equipment pieces, not really. Like most of the things that you would use that get autoclaved, we buy in plastic, so we just like replace it with glass or plastics that can be like re-sterilized.
Okay.
For the shaking incubator, it doesn't require any attachment to like the water or the gas or anything like that. And I had included some pieces in there that help make it more versatile. There's like a magnetic base that lets you put any size like beaker container onto it. It has elastic spring thing, so it can change to hold whatever you want to put on to shake. But those are all like metal components that can be reused. So the typical like bacteria stuff would probably fit in our typical budget. We just replace
Something else
the
that you're not,
consumable
some
stuff.
other experiment
Yeah.
you're not doing, you
Yeah.
have supplies.
And the other piece too is that if because the incubator we would be able to grow more bacteria at a faster rate. We may not need to buy as much initially because we could grow it ourselves too. So it also might save some cost there from year to year. And we'd have to see how much of a savings that is, but I know with the design challenge, we bought some bacteria and we've continued to inoculate more colonies as we've gone through the process, so we're not always buying new bacteria.
I think an honors level elective is a great idea. I think it's something that is a welcome idea. I'm sure the students will be very excited about it. Is this, these plans that you're talking about, I assume that that's really, is that mine? Oh, no, is it just for the honors level class? I mean, I know the pieces could be used in other ways, but these particular plans, are those just for the honors level class, or will there be a non-honors level DNA class? How is
It kind of depends on enrollment, to be honest. We plan on putting the honors option in the course book next year, so kids the following year could take it. So the idea is that we have these components that we could tool to fit the different levels. So both of them could work with synthetic biology and use the bacteria, but one of them might have a more complicated piece, more challenging design your own, or we can find new components related to those that they can play with.
I'm gonna jump in again, sorry. The expectation moving forward with the honors class would be, and I just wanna say this up front, initially it would be that DNA would be an honors level course, it wouldn't be a CP and an honors level course. Again, assuming that the numbers work out so that until we really establish it as an honors level, it would be exceptionally challenging to offer two levels and to make them appropriately different. So initially, we would just sort of forego the CP and replace it with an honors level DNA class, and then as we got solid foundation of an honors level class, we could then differentiate and potentially offer two levels of that course. But this is something that Mary and I had chatted about a bit, to just make sure that it's fair for students and that it offers appropriate rigor, especially given the grade bump and the amount of work that we'd be expecting them to do.
Thank you, Mary.
Thank you. That's great.